Our goal today is to finish this chapter so that you have this material to review over the holiday break. Okay. I know how you guys are. You are studious. That's why you're here. So I give you a lot of credit for that. Let's go ahead and get moving on this. The area that we finished off with was right before here, the intermediate step. They don't consider that part of glycolysis because glycolysis is in the cytoplasm, and this intermediate step goes from the cytoplasm into the mitochondria. So they don't have a really good name for it. And it's not one of the three main steps. So realize that we have 4 steps here, and the one that's not considered a main step is this intermediate step because it moves things from the cytoplasm into the mitochondria. And the thing that it's moving is the pyruvates, which were generated in the cytoplasm, is going to undergo a modification as it is moved into the mitochondria, and the modification is we're going to make an acetyl-CoA or acetyl-CoA. And this is important because we went from a 3-carbon molecule now to a molecule that is actually just 2 carbons being carried by this coenzyme. So this is now 2 carbons in size. And whenever we go down in number of carbon atoms as part of a molecule, we will lose carbon in the form of carbon dioxide. So keep that in mind. And also, whenever we do this process where we go and generate carbon dioxide, we will also generate NADH. NADH and FADH2 are 2 very important molecules because they are carrying the products that we need to put into the electron transport process, which is the last thing that we'll talk about here. Let's move on down to the next process that will occur, the TCA or the Krebs cycle, and that occurs inside the mitochondria in its core, also known as its matrix. An acetyl coenzyme A will enter the TCA. The TCA is a cycle, and it will occur again and again and again as long as you feed it. So as long as you feed it, we'll keep this cycle going. And so the acetyl-CoAs are what we feed into the Krebs cycle or the TCA cycle. For every time we feed in an acetyl-CoA, we will get 1 substrate level ATP. Remember, that's taking a phosphate group off of a molecule and adding it to ADP to make ATP. That's what substrate level phosphorylation is. We will also get 3 NADHs and 1 FADH2, if you want to be a little bit more technical, but as time goes by, you get lazier and lazier, and that's just a symbol of laziness that you actually see there on the paper. And we like these guys. As a matter of fact, of all of these, we like this one because we are going to learn we can get a lot of ATP from an NADH. That's for each turn of the cycle. So what do we need to do when we start off with a glucose molecule? Do we just keep these numbers or do we do something to these numbers here? We should double those numbers, correct? So that's per turn. So for each acetyl-CoA, and we have 2 entering into this cycle. Now, this is where we get the big bang for the buck. The ETP, or the electron transport process, it occurs on the inner mitochondrial membrane. Remember, the mitochondria are double membrane structures, and we are going to find that we have a series of proton pumps. And whenever you hear the word pump, what should you automatically think about? Fuel or ATP, right? We've always in the past used ATP to fuel our pumps because we use pumps to go against a concentration gradient. So what I'm telling you, we are going to pump protons against their concentration gradient. So we're going to go from a low to high with the movement of our protons, and we need energy to do that. And in the past, we've used ATP, but our goal here is to make ATP. And so what we actually are going to use now in this inner mitochondrial membrane as an alternate source of fuel or energy, and that's electrons. Just waiting for you to pop on up. That is just electrons now. So electrons have energy, and we are going to use the energy of these electrons to fuel these proton pumps. So that is very different. The other thing too is what is bringing in the protons and what is bringing in the electrons, and guess what that is? We use NADH plus FADH2. So that's what we're going to use. NADH and FADH2 are going to be our source of hydrogen ions, which are protons, and electrons. And we talked about these guys are the ones that reversibly bind hydrogen and electrons, hydrogen ions more specifically, and the electrons. So NADH and FADH both power the electron transport process, and it says power because remember they're carrying electrons. They also provide the protons for the proton pumps too. Cytoplasmic NADH, which is generated during glycolysis, will use the Malate shuttle, and if they use the Malate shuttle, the electrons will enter into this process at the proton pump called FMN. If we are using the glycerol P or glycerol phosphate shuttle, the electrons are going to enter in and work on fueling this process at CoQ10. CoQ10 is also an enzyme. Some people take that as an over-the-counter supplement. Oxygen will be the final electron acceptor. So one of the things about this process is that electrons, energy is neither created or destroyed. Correct? You guys learned that a long time ago. So electrons are going to share their energy, but they are not going to be destroyed. And so what's going to be interesting about these proton pumps, the electron on one is going to be transferred to the next proton pump, which it will fuel, which will then be transferred to the next proton pump, which it will fuel, and the electrons will finally be transferred to actually an oxygen atom with the presence of a couple of protons to make water. So that's one of the reasons why water is part of that chemical reaction that we see when we talk about the breakdown of glucose. And so oxygen is the final electron acceptor. That's why we call it oxidative phosphorylation, and we end up with water because of that. This is something that you'll have to memorize, all of this. And the memorization can seem a little bit intimidating at first unless you break it down. And so it's very important that you actually work on breaking it down. So let's add a page here and let's go over how we are going to break this down. We'll start with the first step, which is glycolysis. So we'll start with glucose. We know that's a 6-carbon molecule, and you just write this over and over again until you have it memorized. And so we're going to have a split here, and we're going to make DHAP and G3P. DHAP is readily converted to G3P. G3P is what's going to make our pyruvate. DHAP is 3-carbon, G3P is 3-carbon, and pyruvate is 3-carbon. Now, remember I said this is a 10-step process. This is the only steps I want you to memorize. But in addition to these steps, you have to know a couple of things about what's going to happen at these areas of the arrows. And in order to make our ATP or money, we're going to have to put in a little bit of ATP or money to actually create a bunch. And so we are going to start off using 2 ATP to actually get this wheel or process going, and we'll get 2 ADP. The other thing that I want you to do, when we make G3P, we will end up with ADP being converted into ATP, and we'll have some NAD being converted into NADH. Ultimately, how many G3Ps are we going to funnel into this pathway? How many? 2. So let's go ahead and right now, because this is the basic reaction, and now I'm going to put 2 G3Ps because we've acknowledged DHAP is going to become a G3P. And with each G3P, we would get 2 ATPs. So since we're having 2 G3Ps, 4 and 4. You guys working with me on that? So that's because we're dealing with two G3Ps. And as far as NADs, we are going to actually use two NADs, so it's basically one per G3P, and get two NADHs. So that's one step you have to memorize. And these guys, because we're going to actually count up ATPs, NADHs, and FADHs. Then, so that's glycolysis. Where does this occur? It occurs in the cytoplasm of the cell. Now, pyruvate has to get into the mitochondria, and it will be groomed, so to speak. So it will be groomed and it will be changed. And that's that intermediate step. And so we will move it into our mitochondria. And when we move it into the mitochondria, it will go from pyruvate, which is 3-carbon, to acetyl-CoA, coenzyme A. And we're going to get 2 pyruvates, correct, in this whole process, right? So how many acetyl-CoAs will we get? We will get 2 of those. But acetyl-CoA is 2 carbons. And so that means that little story I told you about whenever we get carbon dioxide, we always get NADH. So it's important that you actually put this into our equation too. So we're going to get CO2 in this process, and obviously that will be 2 for each pyruvate, and we will have NAD converted into NADH, and there will be 2 for each acetyl-CoA that we produce. Now, acetyl-CoA is the one that we're going to feed into the TCA or Krebs cycle. And this is how I memorize it. You guys can memorize it in a different way if it works for you. But acetyl-CoA will combine with a 4-carbon molecule that's called OAA, oxaloacetic acid. And so we will combine a 2-carbon chunk with a 4-carbon chunk, and that obviously will give you a 6-carbon chunk. And that's called citric acid. So those are the 2 names that I want you to learn. But then after that, there isn't any other name I want you to learn other than the rest of the cycle and the number of carbons associated with the products of the rest of the cycle. So we're going to do another 6C. We're going to do a single 5C. We're going to do a 4C, which is going to go to another 4C, which is going to go to another 4C, which will end up with oxaloacetic acid. So there's 4 4s, 2 6s, and 1 5. The only names that you need to know is this one and this one as far as the names of this. And the other thing that you need to do is whenever you see that we lose a carbon dioxide because we went from a lower number, CO2, I want you to go ahead and make sure that we have an NAD going to an NADH. So it's twice right now. So that's easy enough. So automatically, whenever there is a carbon dioxide generated, you need to go ahead and put that in the cycle, and then you need to go ahead and make NADH. Now, the next step is going to be the substrate level phosphorylation, and we will have GDP, guanine diphosphate, making GTP, guanine triphosphate, and GTP is going to interact with ADP to make ATP. I'm just getting really lazy there on that. It's actually in that diagram. The next 4 to 4, it's a conformational change. And with this conformational change, we actually now find FAD turning into FADH2. And then we're going to have one more NADH formed, and it's the very last 4 to 4 NAD is going to be NADH. So indeed, if we count up our NADHs that we get, we have 1, 2, 3 NADHs, 1 FADH2, and 1 ATP per turn of this TCA or Krebs cycle. We do this twice, and so these numbers have to be doubled if we're doing what I call an ATP balance sheet, which we're going to be doing in just a little bit. So now let's go ahead and go back into our notes and take a look at the next thing that I want you to be able to draw on your own. You can easily draw your outer mitochondrial membrane and your inner mitochondrial membrane. I'm going to just do this off to the side. So outer, inner, and inter. This is our intermembrane space. And we're going to have in this 4 transmembrane proteins on the inner mitochondrial membrane. So I'm going to make one block. I'm going to make another block. I'm going to make another block. And I'm going to make this funny looking Erlenmeyer flask kind of a guy. Those are transmembrane proteins. And the first 3 are pumps, typical proton pumps. And so we're going to have protons in the mitochondrial matrix that are going to be pumped through these proton pumps. And the name of the first proton pump where everything starts is called FMN. The next proton pump is BC1. And the next proton pump is AA3. There is this protein that is embedded in the inner mitochondrial membrane called Q. Q is important, and so let's make sure you add that in. There's another one, but it's not as important as Q. And what's going to happen is NADH is going to donate its proton and its electron to fuel this pump at FMN. In the book, sometimes you'll see this listed as 1 proton pump. What's important is how many protons are pumped by FMN per open door, so to speak, and it's 4. So we have a bunch of protons in the matrix of the mitochondria. Here comes an electron, and he's like your hand on a button to open a door. And as long as that electron is on that hand of the button, it will open the door and allow 4 protons to be pumped in. And the door will close. Even if the electron is still standing right there pushing that button only will open and allow 4 protons in, and the electron has to then leave. Because if the electron doesn't leave, we can't pump anymore. Okay, you guys follow me? So then the electron leaves, and that's why you actually see down here, you see some of that story. The electron is going to go to Q. It's the same electron that was on FMN, but it goes to Q, and Q is just an electron carrier. It isn't going to pump anything. It just happens to be a place to put the electrons so that we can get them off of FMN. We want the electrons to come there, do their business, and then leave. because if they don't leave, we can't use the pump anymore. Okay, so it's passed off to Q, and then Q, which isn't a pump, is going to pass it off to BC1, because BC1 is also going to pump 4 protons to the intermembrane space. So that electron's pushing that button, the door opens and allows 4 protons, or pulls, however you want to do it, 4 protons into the intermembrane space, and then the door closes. But the electron has to move off, or else we can't use this pump again. So then, as our story shows us over here, the electron then gets moved over to AA3. So here was our electron being transferred over to BC1. We pumped our 4 through, and then electron gets transferred over to AA3. AA3 is a little different from FMN or BC1 because it's only 2 protons that get pumped into the intermembrane space. And again, we have to get rid of those electrons because the pump won't work unless we supply it with a fresh batch of a fresh electron. And so what happens here is that electron gets transferred to an oxygen atom, which will then combine with 2 protons and make H2O. And so the electron, remember, electrons are part of bonds. And so the electron is where the electron is actually very happy to be. It's actually going to be part of those bonds, those covalent bonds in water. Now, the next protein, transmembrane protein that's in here is ATP synthase. And like the name, it makes ATP. And what it does is we will have ADP plus P little I. And that's also on the other side too, if you want to see it written a little bit more beautiful. Obviously, somebody took their time to actually draw this diagram out, and is much nicer in their handwriting on regular paper than I am on this symposium. But we cannot make ATP from ADP and inorganic phosphate. And you guys remember, inorganic phosphate means that it's not attached to any molecule that's carrying, that is composed of carbon, right? That's what inorganic means, because if you're organic, you've got carbon as part of your structure. So inorganic phosphate is hanging out there. And what we'll do is we will use this proton gradient that we've created to actually generate power of this, what I call like water wheel, to actually generate the energy to make ATP from ADP and inorganic phosphate. And we need 4 protons to go through this ATP synthase. And so I'll put H, oh my gosh, handwriting gets worse as the day progresses, H plus. And your notes are odd here. I don't know what happened. Everybody has a brain fart. This should be 4 in that drawing there. It shouldn't be 2. 4 protons in are going to give us 4 protons out. Energy and matter is neither created nor destroyed. We're not going to destroy it here. So with this process, we are putting in with an NADH, with one NADH, we are able to have how many protons in the intermembrane space with one NADH? 10. And it takes only good Lord, takes 4 protons to make 1 ATP, as we just saw this right up here. 4 protons to make ATP. So what's the technical number of ATPs that we generate from one NADH? 2.5. Okay, so that's the reason why NADHs are so good. NADH will give us 2.5 ATPs per NADH. The other story about the other cofactor that's carrying protons and electrons is FADH2. And FADH2 doesn't actually deliver its products to FMN. It delivers its material and electrons to Q. So that's what we actually see here. So protons are left off in the cytoplasm, or matrix. I shouldn't say cytoplasm because you might think of cell. The matrix of the mitochondria. And the electron that FADH2 is carrying is given to Q. Now remember, electrons are fuel. Q doesn't do anything, so it's going to pass the electron right off to BC1, and of course then the electron will go to AA3, and ultimately it will go to oxygen, and we'll end up again with water, the oxidative phosphorylation stuff, because that's the oxygen aspect of this. So we have to have oxygen there. Now, let's say we have no oxygen, because the electron has to be handed off to that oxygen atom to give us a water molecule. Say we have no oxygen. We've run completely out of oxygen, which happens when we die, correct? Okay. So what that means that you have no place to deposit your electrons. So what does that mean to AA3's ability to pump more protons through it? It can't, because remember I said the electrons go, they hit the button, the door opens, and 2 protons are allowed through, and then the door closes. And we can't do this unless those electrons leave and another electron or electrons come in and hit the button again. So if I can't get rid of my electrons, they stay here at AA3, and they make the pump ineffective, correct? Well, if I can't get rid of my electrons at AA3, what about the electrons that were at BC1? They're going to stay there too. So BC1 is rendered ineffective. Again, if BC1 is rendered ineffective, then Q can't pass its electrons, and therefore FMN won't work either. So oxygen has to be present in order to make ATP with this process, because we have to actually move the electrons along if we are going to actually use these pumps. So that's an important aspect of this. One of the things that happens is we can actually poison this process. There are poisons out there that will poison this process, and if they poison this process, what does it do to your life? It terminates your life, because the bulk of your ATP is produced here. Remember, our mitochondria are little powerhouses of the cell. And so if you poison the pump of the powerhouse of the cell, you've poisoned yourself. You've poisoned the cell. And you'll have cellular death and organ death, and then system death, and then organism death. There's an interesting poison out there that blocks our ability. Let me erase all this stuff. It's a nice thing about this. I can do a lot of erasing. There is a chemical out there that blocks this very last step right here, where we actually have the electrons go to oxygen and make water from this process. And it's called HCN. Do you guys know what HCN is? That's cyanide. This is where cyanide works, Sonia. Cyanide kills you. You've heard of people trying to off their spouses and stuff like that with cyanide poisoning. So cyanide kills you because it irreversibly hits this portion of the electron transport process, and you cannot make any more ATP in that mitochondria. And so that's where it works. That's where cyanide is so lethal to the individual. It causes your mitochondria effectively to become non-functional. Now, it would be great if we could actually put another proton pump in here. And let's see if we put another proton pump. Let's put it right over here, and let's call it ZAO. I can name it after myself, right? I discovered it, so I named it after myself. So if this proton pump acted like FMN, then that means 4 would go through and 4 more would be here. And then how many ATPs could we potentially generate now that we have this additional proton pump? 3.5. Okay? But if if my ZAO pump acts like AA3, it would be 3, correct? Because that's only 2 protons. You guys following how to do the math here? So we need 4 protons to generate 1 ATP. And this intermembrane space to go through ATP synthase. Now, what if I told you that I misidentified the location of my ZAO pump in this mitochondrial membrane? And it's actually over here. Would this ZAO pump generate more ATP or not if it was there? Not. Because what's important is where we actually start with NADH and FADH. If I didn't say that the ZAO pump was preceding the FMN pump, if I said that and I didn't say that now the NADHs will drop off their electrons at the ZAO pump, you have to understand that this ZAO pump position is very important in the generation of more protons in the intermembrane space, because this ZAO pump requires electrons, but it's not where electrons are in the chain of where electrons are dropped off. You guys follow that process? So wherever a pump is theoretically placed for an exam purpose it will indicate whether or not we'll make more ATP or not, and how much ATP, depending upon whether the ZAO pump acts like an FMN pump or an AA3 pump, correct? So that's something to keep in mind as you look at old exams. Let's take a look at the ATP balance sheet.
What if that ZAO pump is after that AA3?
If it was after AA3, then because it's after AA3, the electrons would be transferred, correct? And then ultimately then the electron would hopefully be transferred to oxygen molecule or atom and make H2O. Yes?
In our diagram, there's only one Q person, right?
Correct.
Okay.
Yeah. Q is not a pump, it's just an electron carrier. So it has nothing to do with that. And you guys know where the hydrogen's coming from, right? It's coming from NADHs and FADH2s. So that's where the hydrogens are coming from, and the electrons are also coming from those. And that's why in the beginning of our notes, it was important that you realize that when we're carrying hydrogen, we're also carrying electrons. It's the whole part of that process with those cofactors. Looking at the ATP balance sheet, glycolysis, remember that's in the cytoplasm of the cell. We have 2 ATP entering the glycolysis process, so we're going to give them a negative number because that's less than what we're making. We end up though with 4 substrate level ATPs, and remember we end up with 2 NADHs. And as far as the NADHs, let's go up and take a look at our notes here. If the malate shuttle is used, the electrons go in at FMN, which means we get the normal production of ATP. But if the mitochondria, for some darn reason, have a glycerol P or glycerol phosphate shuttle, those electrons are actually delivered to Q, which means you lost the protons that you got from the FMN pump. So you'll end up, because you lost 4, instead of 2.5, you'll get 1.5 ATP out of that process. In other words, it's acting like it was FADH if you use a glycerol phosphate shuttle. So that's why in our notes here, we have 2 NADH that go to the electron transport, and we get 3 if we're using the glycerol P shuttle, or 5 if we're using the malate shuttle. So total for the glycolysis is 5 at the worst, 7 at the best. The intermediate step, we end up with 2 NADH out of that process for 1 glucose molecule, and we're in the mitochondria, and so we'll end up with 5 ATP. In the TCA cycle, we already talked about these numbers here, and multiply those by 2. And remember, for each NADH, we get 2.5 ATP. So for 3 NADHs, therefore we get 7.5 ATP. And we double that because we're taking 2 turns of the cycle for each glucose that we actually chew up, so to speak. So that's why we get 15, 3, and 2. So out of the TCA or Krebs cycle, we'll end up with 20 ATP, and we get a grand total of 30 if we use the glycerol P shuttle, or 32 if we use the malate shuttle on those NADHs that were generated in the cytoplasm of the cell. So that's the major biochemistry chart that we wanted to go over. Now let's go over lipid metabolism. Okay, lipolysis, lipid oxidation, and of triglycerides. What's important is the triglycerides are broken down into glycerol and fatty acids. It's important to realize that actually triglycerides are the major fuel for your skeletal and cardiac muscles and liver. So in other words, if you showed them glucose and you showed them triglycerides, you go, oh, I want the fat. I want the triglycerides for my fuel. That's my preferred fuel, which is good because other cells are going to be the ones that, like the brain, that want glucose, which the brain is a preferential glucose feeder. Okay. So remember, glycerol is a 3-carbon chain, and we're going to have 3 fatty acids attached to it. And when we break it down, we'll break these bonds and we'll get glycerol plus those fatty acids. I don't know if you remember, but there's something very important you need to realize about the carbon length of each fatty acid chain in our body that occurs naturally. Do you guys remember what's really important about the number of carbons in our fatty acid chains? They're always an even number. So our fatty acid chains are always an even number of carbons. And this is important because let's take a look further on down at the notes. Glycerol. Well, when we break down triglycerides into the glycerol and the fatty acid chains, glycerol gets converted to G3P. You guys know where G3P is in the cycle. It's way up there in glycolysis right before we make pyruvate. So that's an important thing to realize. Fatty acids, the important thing about fatty acids being an even number of carbons, which is 2, by groups of 2, fatty acids are use a process called beta oxidation and are converted into a bunch of acetyl groups. So if you have a fatty acid chain that's 20 carbons long, how many acetyl groups will you get? 10, right? 10 times 2 is 20. Okay, so that's an important thing, and you have 3 of those. And where do the acetyl groups go in? Into acetyl coenzyme A, which feeds right on into the TCA or Krebs cycle. In glucose starvation, what happens is we reverse the cycle. We will take oxaloacetic acid, which is also oxaloacetate, and we will convert it into glucose. So we will rob the Krebs cycle of OAA. If you rob the Krebs cycle of OAA, what happens to the cycle? It stops. So that's an important thing. We are then interfering with the Krebs cycle whenever we do this. And so we will have then TCA slows down and stops. And we will have acetyl-CoA accumulating. And if acetyl-CoA is accumulating, it's not going anywhere. We've got to do something with it. It gets shipped to the liver, because the liver is a major organ that helps us with detoxification and purification and stuff like that. It will get shipped to the liver and will create ketones. And this will cause an acidosis. Because even though the liver is trying to process it, it doesn't process it very well. It makes ketones and diabetic ketoacidosis. Isn't that what glucose starvation is actually all about? So DKA is telling you that we are going to rob the Krebs cycle or the TCA cycle of OAA, therefore make that cycle ineffective, because now we're trying to get our energy from somewhere. Okay, and OAA is going to help us to reverse engineer our glucose, but because we are now using triglycerides as a fuel, we are basically getting this buildup of acetyl-CoA, and we can't feed it into the cycle anymore. And so we have to do something with this buildup of this acetyl-CoA. So the liver will take it, is trying to do something with it, makes it ketones, and ketones are acidotic, and we get diabetic ketoacidosis. And that's the whole process there. And if you have diabetic ketoacidosis, how are you trying to normalize your pH? Hyperventilate. You're trying to blow off CO2 to cause an alkalosis, trying to actually buffer that acidosis, and it works at first, but doesn't work for the long term. Lipogenesis, we're all familiar with that, aren't we? The formation of our own fat. And that's because when we take in too much fuel, or glucose, which is what the precursor is to generation of ATP, we will have excess ATP causing a negative feedback on its own production. Well, that makes sense, you don't need ATP, so let's stop making ATP because we got plenty. And what happens is we'll end up with glycerol being produced from G3P, by the way, and we'll end up with some fatty acid chains because of reversive beta oxidation actually going on. Because if we're having negative feedback, everything is building up backwards from there. But this will, in the presence of glycerol, the fatty acids will form from acetyl-CoA, and we'll get our lovely TG, our triglyceride, which we know is what our little fat cells love to actually hold inside their little homes. Protein metabolism. Amino acids, we know, are the building blocks of proteins, and we only make a protein if we have all the amino acids present, right? Because a protein is very dependent upon the proper sequence of amino acids. So proteins require proper types, and obviously number, of AAs, amino acids, to form. If we don't have the amino acids present, we won't make the proteins. Because that's just the way it is. The protein chain is very specific for the amino acids. And if we don't have the proper amino acids to make the proteins, then what we'll do is that we will oxidize our amino acids for energy. Excess or leftover. But you realize that's not the first thing that we want to do. The first source of fuel that we want is glucose, and the second source of fuel that our body will actually go after is triglycerides or lipids. And so we don't want to really utilize protein for our fuel because that means we're actually kind of deconstructing ourselves whenever we do that. And for energy, if energy needs are not being met. So this process actually only occurs as a last resort. So with protein oxidation, we have two major steps, deamination and transamination. Deamination, just like the name sounds, the amine group is removed and it is converted into urea in the kidneys. The remaining components will go into cellular oxidation for energy purposes. Transamination, amino acids are converted from one form to another, and this is actually really nice. Liver helps us do this transamination process, and we do this because it's important that we actually have the proper building blocks for protein synthesis. Quickly, I just want to end this material for our day-to-day, so we might go like 2 minutes over, but it won't take us much time. Role of the liver in metabolism. The liver will actually, glucose is the main monosaccharide we like, and it's interesting, the glucose takes fructose, because a lot of people say you can't, digest, you can't utilize fructose, but you can. Fructose and galactose are converted into glucose in the liver. So the preference is glucose, and the liver will take fructose and galactose and convert them into glucose. The liver itself actually produces cholesterol, and approximately 85% of your blood cholesterol comes from the liver. And so when you get your blood cholesterol levels checked and they're too high, it's actually mostly the liver that is doing this because of something that you may be inadvertently doing dietarily for yourself. But that's what the liver does. It makes your cholesterol. The liver is responsible for the packaging of fatty acids for transportation in the bloodstream. It synthesizes plasma proteins. You guys remember some plasma proteins that we've talked about before? Complement, yes, that's where it is generated, and clotting factors. Oops. indeed forms and processes amino acids. So again, that's the deamination, transamination. Stores and releases glucose. Storage form of glucose, you guys know, is glycogen. It's our major detoxification organ for things like alcohol and Tylenol, and also creates lipoproteins, lipolipid proteins, and cholesterol is all wrapped up for transportation in a very important package by the liver. So lipoprotein is used to transport our fatty acids and cholesterol in our bloodstream, and we have 3 of them that are measured on a relatively regular basis in our society. HDL, it stands for high density lipoprotein. LDL stands for low density lipoprotein. So of these 2, one floats better than the other. Which one of these 2 will float better than the other? LDL, low density, which means I'm lighter and I'm fluffier, lipoprotein. High density lipoprotein is called that, and it has a tendency to sink if you're actually trying to stratify it out of the blood. And then there's VLDL. V stands for very low density lipoprotein, and it's the most floatiest and fluffiest of them. HDL is interesting. It is the good cholesterol. Most people call it the good cholesterol because it's picking up cholesterol and returning it to the liver for disposal. So HDL is good cholesterol or happy cholesterol or however you want to. It's the one that's cleaning the graffiti off the walls. I consider cholesterol graffiti on your walls. LDL is designed to actually move cholesterol to our tissues to be deposited or used in our tissues. So that's what LDL is. And if you have a high LDL, that means we're putting all this cholesterol out there and we're putting it more out there than what you need. That's what LDL is all about. We deliver cholesterol. We deliver it, we deliver it, we deliver it. VLDL is called that because the very low density lipoprotein, it is triglycerides that it's really full of. And it is designed to take triglycerides to your fat cells for deposition there. When VLDL drops off the triglycerides, it becomes LDL. So that is the cholesterol story, and I think we're going to end right there. You guys have any quick questions? Yes.
Can you bring up that page you drew one more time, please?
Which?
The one that you drew.
This, oh, the one that I was drawing? Yeah. All you need to do in memorizing these steps, because I like to go in and talk about each step, is start with glycolysis. Start with this first one here and just write it over and over till you memorize it. Then go to this guy here and write it over and over till you memorize it. Break it down into digestible chunks. Chunks. I need coffee. I really need coffee. I got up at 5:30 this morning. I really need coffee. I am not a morning person. Okay, this step here, it's 4 to 6 to 6 to 5, and then make sure there's four carbons in this. And then figure out where your NADHs and CO2s are, because that's the easiest, and then memorize the rest. Okay. Yes?
Could you go to the lipid metabolism page?
The lipid metabolism? So oxidation is breaking lipid down, so it's a catabolic reaction, and lipogenesis is an anabolic reaction. And so remember, we have the glycerol backbone and 3 fatty acids, that's why it's called a triglyceride, 3 fatty acid chains attached. And each fatty acid chain is even number of carbons. And that's very important. That's the kind of fatty acids we make and we utilize. And that's very important because then beta oxidation breaks it down into 2 carbon chunks, which is fed into the acetyl-CoA portion and then forms with OAA to make citric acid, and we got the TCA cycle running at high speed because we get a lot of 2 carbon chunks out of this guy, right? So. And if you have run out of carbs or glucose and are therefore using this, that's where the glucose starvation story feeds in, because the brain is an obligate glucose feeder, and we need to have glucose for the brain, and it will rob the TCA cycle of OAA to make glucose. Once you go over it more and more, then it becomes more familiar. Right now, it's a lot of biochemistry. Yes? Yeah. Okay. So, you guys have a wonderful spring break. Yay! We're done with this chapter. And then we'll do a clicker, which you are obviously very much waiting on. See, I'm just excited.